Okay, so when you click on that, obviously it opens up, same as always. Somebody put tape on my smartboard. Why? Oh, that makes me angry. Okay, so yours is going to look like this. Okay, we're going to be going over kind of how you're going to write up the problem and what an abstract is, talk about your variables, that kind of stuff should all be familiar. Okay, and we'll get you going on that here as soon as possible. Okay, so the idea here, guys, is we're reviewing two things with this activity. We're reviewing experimental design / scientific method, okay? And then we're reviewing the factors that affect plant growth. So what you're going to be doing is designing an experiment, not running it, just designing it, okay, to test the effect of a variable you choose, okay, some factor affecting plant growth, on the growth of a plant. What? So you're going to have this experiment that you're going to design. So a couple of reminders about a proper experiment. First off, you have to have a problem that you're investigating. So that's going to be fairly simple. What is the effect of whatever your variable is going to be, whatever your factor that you're choosing is going to be on plant growth? Okay, that'll be it. Then you're going to have your design. So what are your variables, right? Well, whatever factor it is that you're testing is going to be which variable? Everybody's responding variable will be the same. It's the right, the growth of the plant. Okay, the manipulated variable is going to be whatever factor you've chosen. All right, so like I've seen people who do this experiment, and one person, this was like the weirdest one, I think. They were going to water their plant with different liquids. Okay, so they were saying, what is the effect of different liquids on the growth of a plant was their problem. Okay, and then their manipulated variable was that plants require certain nutrients and whatever to be in the soil. So instead of watering with just water, some plants will be watered with like Gatorade, coffee, hydrochloric acid. I don't remember what but they did a bunch of different things, and that was their experiment. Okay, their manipulated variable was the material or the liquid they were watering with. Now, the thing you have to remember is you can only have one manipulated variable. Okay, that's where your controlled variables come in. You have to control everything else. Any other factor that affects the growth of a plant needs to be the same for all of your plants. Does everybody follow me? Okay. Otherwise, you're not going to know whether the thing you changed caused the plants to grow differently or whether something you forgot to control caused them to grow differently, right? Like I once had a student who did this lab and they wanted to do the effect of light on plant growth. And so they put one plant in the sun and they put one plant in a closet, right? Well, what else is different between a closet and being in the sun? Temperature, okay? If you sit something in the sun, it's going to be much warmer than it is in the closet. Now, I mean, granted, that's a pretty simple experiment, but I mean, also putting a plant in the dark is going to kill it. But you get the idea here. They didn't control all the variables that could affect the growth of a plant. So you need to keep those things in mind, okay? And that's kind of what part of your mark will be on this is how well did you remember all the factors that affect plant growth and how well did you control them? Okay, everybody with me there? Right, so it'll be same as always. You'll have one manipulated variable. Make sure you only have one. Okay, and that's what I mean by that person who put the plant in the closet and the plant in the sun. Okay, they actually had two manipulated variables, darkness and temperature, and they didn't realize that. Okay, so you have your variables, then you'll have your hypothesis. Okay, your hypothesis will work same as it has on every other lab. If, and the if part's going to talk about what you're testing. Okay, if this has an effect or has this effect on plant growth, and we do this, then okay, and the then part will describe what you're going to see. Okay. Everybody with me on that one? All right. Then you're going to have your procedure. Okay. Now the procedure is where you are going to tell people exactly how you are going to control all of your controlled variables. Okay, you can't just say in the design, I'm going to control all these variables. In the procedure, you have to say, here's how you're going to do that, right? So, if you're growing these plants all from a seed, that's good. Make sure they're all from a seed. Don't have one that starts out as an adult, one that starts as a seed, and one that starts as a juvenile. It's not a fair comparison, okay? Things like how much soil, what kind of soil, like tell them how you're going to do that. Okay, how exactly are you setting this up? How much of this are you using? Those are details that need to be in the procedure because they're part of the setup. The other stuff that needs to be in the procedure is how are you going to measure plant growth? There's a lot of different ways to do that. Okay, and you might want to use more than one. Plant growth is pretty general. Okay, if we're talking about like you're doing this experiment on tomatoes, one of the measures of plant growth could be height of the plant. Another measure could be how many tomatoes does it produce? How many blossoms does it get? Okay, there's all kinds of ways to measure the growth of a plant, and you might want to use more than one. So in your procedure, you need to describe to the person running your experiment, here are the measures of plant growth. Here's how to measure them. Okay, so you're basically telling them how to collect their data from your experiment. That's part of experimental design, is how are you going to collect your data. Okay. Making sense? That's as far as it goes. Okay, you're not going to have analysis. You're not going to have data because you're not running this experiment. You're just designing it. So it ends at procedure. Okay, everybody with me on that? Okay, so you don't have to go any further than that. Now, the thing that will be different here, that's actually going to come right at the very beginning, I skipped it, okay, is what's called an abstract. Okay, an abstract is basically a quick summary of your experiment as well as why it should be run. In real science costs money. So if people are going to do research on something, they usually have to get a grant for it. Okay, and a grant means some company somewhere pays them to do the research, and then when the research is done, the company effectively owns the results. Okay, unfortunately, that's how science works. Scientists very rarely get rich on their own discoveries. Okay, because whoever's funded it, okay, gets to use it, and that's like pharmaceuticals would be like the prime one. Okay, you develop a cure for cancer, you're still going to be poor, but you'll have a good feeling inside. Okay, but the pharmaceutical company will get filthy, stinking rich on it. Okay? That's how science works. So your abstract has to be a little bit of a sales pitch. You have to explain to people, here's what I know about plant growth. This is what I'm going to test. And these are some of the applications of my research. How could this benefit people? How could it be used to be profitable or something like that? Okay, now most of the stuff to do with plant growth could have some agricultural implications, feeding, maybe more people or something like that. That's stuff you have to put in your abstract. So it should be about 3 or 4 sentences. Okay, here's what I know about plant growth. This is what I'm going to test, and here are the practical applications. That's probably 2 sentences. Okay, so you start out with your abstract, then your problem, variables, hypothesis, procedure, you're done. Okay, questions on the expectations here? All right, so remember that your procedure needs to be detailed. Okay, you got to explain every little thing. How am I going to set this up? I'm going to get pots for the plants that are this big, or I'm planting them all in this place in a garden, or whatever it is you're going to do. Explain each and every little nitpicky detail about how you set up this experiment. It has to be that detailed. Okay, that's going to be the biggest part of this whole thing, is the procedure. Okay. Yeah, I think that covers it. So you're going to be the places where you'll get marks for the design. Okay, you'll get marks for the hypothesis. Part of the design is also your abstract. Okay, so and then 5 marks for the hypothesis, 5 marks for your procedure. Okay, and 5 marks for overall. So you'll see there's no analysis, there's no conclusion, there's no evaluation. Right, so those parts are not on here. Okay, you have all class today to work on that. Okay, but it needs to be submitted at the end of class. Okay. All right, if there's questions while you're working, just come on up and ask.